So, from today we are going to learn about secondary metabolism in plant cells. Now comes the commercial applications of plant cell technology. So, before we move on to how we exploit secondary metabolism commercially, it is important for us to know about secondary metabolism in plants, its role, and hence we will take it extrapolated to commercial applications of it. So, what is secondary metabolism? Now, what is metabolism? To begin with, it is the sum of all chemical reactions that take place in an organism. So, this forms metabolism, whether primary or secondary. Now, most of the carbon, nitrogen or energy which is accumulated in the cell is majorly used for its growth and development. So, that forms the primary metabolism. Now, some of the carbon and energy is also utilized for specialized functions, which majorly deal with more organized function and differentiation in plants, which deals with its survival in the nature for competition or defense related, all that. So, most of the carbon, nitrogen and energy, it ends up in molecules required for proper functioning of the cells and organelles. Now, for example, what are those? We know lipids, carbohydrates, nucleic acids. Now, these form the primary metabolites. Now, most plants divert a significant proportion of this assimilated carbon pool and energy to synthesize organic molecules. So, these secondary metabolites are generally organic compounds. So, these organic compounds actually have no obvious role in general plant growth and development, but they have other roles which come under secondary metabolism. Okay. So, how are they different? At the biosynthetic level, primary and secondary metabolites, generally there are common pathways. Majorly you will find what all pathways in primary metabolism do we know about? Pentose phosphate pathway, glycolytic pathway, then TCA cycle. So, most of the secondary metabolism also leads from some one or more of these intermediates of the primary metabolism, and then it gets diverted into secondary metabolite pathways. So, at the biosynthetic level, primary and secondary metabolites, they share many of the same intermediates. Now, they are derived from the same core metabolic pathways which we know as a part of primary metabolism. Now, secondary metabolites generally occur in relatively low amounts in comparison to primary metabolites. What can be the reason? Then I will ask why they are formed in the stationary phase. So, now try to reason out, why do you think they will be produced in small amounts? So, two things. One is only when it is needed, the cell would spend its carbon pool and energy in this biosynthesis, and only up to a desired level it will try to use the. So, every time the cell would like to conserve its carbon and energy pool. So, only therefore you will generally observe that secondary metabolites, first they are low volume, which means they are present in very low quantities in the plants. The second thing is there is a wide array. You can't even imagine how wide is the secondary metabolism in plants. It is not uniform. It can vary from plant to plant in terms of genus, in terms of starting from family, then genus, even till species you can find variations in secondary metabolites. So, for every such metabolites there will be a carbon required, carbon flux. So, there is a wide array. So, the cell would like to judiciously use and prioritize. So, secondary metabolites generally occur in relatively low quantities, I said. Then their production is widespread or restricted to particular families, genera and even species. Now, what is the role of secondary metabolites in plants? Now, secondary metabolites, they have ecological functions in plants. So, what kind of functions do they carry out? So, they prevent the plants being eaten by herbivores, or they prevent the plant from being attacked by pathogens. These pathogens can be fungal pathogens, can be means of microbial origin, can be insects, can be predators, or like herbivores, can be mammals, can be humans. So, they need to have that wide spectrum of defense against all these, and then they also need to prevent against environmental stresses. So, they serve as attractants, and it is not only defense, but they also carry out higher order functions, like for example in pollinators, attracting the pollinators, or even attracting the predators of the pathogens. So, by using these volatile secondary metabolites which can attract these predators so that they can get rid of the pathogens. Then they serve as attractants like in the form of odor, color, taste for pollinators and seed dispersal. Then sometimes in order to prevent being eaten up, you will find some of the fruits are very bitter, so that humans do not eat. Now they function as agents of plant-plant competition. Sometimes you will see that plants produce exudates in the soil which are toxic to the nearing weeds or the nearing plants. This is a kind of overcoming the competition for the sunlight and for the resources in the soil. So they increase the reproductive fitness. How do they increase? By warding off fungi so that they remain healthy, then bacteria, whatever pathogens may attack them, so that the status of the plant is quite healthy. So the ability of plant to compete and survive is therefore profoundly affected by the ecological functions of their secondary metabolites. This is the general class. There are many other different subclasses of secondary metabolites, but majority of them start from these three major classes of secondary metabolites in plants. What are these three major classes? One is terpenes, then phenolics and nitrogen containing compounds. Your high value compounds which are alkaloids, your anti-cancer agents, Taxol and all these, is they or camptothecin or vincristine, vinblastine, they come under alkaloids, and these alkaloids are nitrogen containing compounds. So this is a general picture which shows that it starts from primary metabolites, primary metabolism, and then some of these intermediates get diverted towards the formation of secondary metabolites. So you will find that shikimic acid pathway or pentose phosphate pathway or MEP pathways, they may independently produce some of these secondary metabolites, or some of these intermediates from two different pathways can come together to give rise to the new secondary metabolism pathway, biosynthesis pathway. So you will find different types of secondary metabolites: coumarins, anthocyanins, flavonoids, alkaloids, terpenes. So there are n numbers. So let us talk about terpenes. Now what are terpenes? They constitute the largest group of secondary metabolites present in plants. Now one of the most popular biopesticide, which is azadirachtin, I gave an example also, is a sesquiterpenoid. So its structure comes from terpene, and then it's a sesqui means there are because of the structure, the number of carbons and the number of rings involved, the terpenes and then its derivatives, it is formed. So the terpenes or terpenoids, they constitute of the largest class of secondary metabolites in plants. Now most of the diverse substances of this class are insoluble in water. So they are not soluble in water. Then certain terpenes are well characterized, have well characterized function in the plant growth or development, and so can be considered as primary rather than secondary. So some of these terpenes also can play a role in the growth and development. So therefore they are classed as primary rather than secondary. For example, some of the plant growth hormones like gibberellins. Gibberellin we have studied, it is one of the plant growth hormone. So gibberellins are an important group of plant hormones, and it is a diterpene moiety. Now bacinosteroids, another class of plant hormones with growth regulating function. So it takes care of the primary aspect of the plant growth and development. So it is also termed as primary metabolite, but it originates from triterpenes. So generally you will observe that most of these high value terpenes, they are plant defense related triterpenoids or terpene molecules. Now terpenes, they act as toxins and feeding deterrents to many herbivores, insect and mammals. That is the very reason why azadirachtin is used as a biopesticide. It acts as a feeding deterrent to the insect pest. Monoterpene, it also depends on the mode of action. Suppose if it is a wide spectrum of, like azadirachtin has a wide spectrum of antifeedant activity. So it can cater to a large range of insect pests. So that is the reason why neem is being used as against even mosquitoes. Then it is also used for pathogens or for biopesticide in crops to remove insects, pests. So it has a wide range of applications. Now monoterpene esters are also called as pyrethroids. This is also a class of biopesticides found in the leaves and flowers of chrysanthemum species, which shows insecticidal behavior. So it is also, apart from azadirachtins, it is also used, or azadirachtin related liminoids, it is also used as biopesticides. Now you can see and make out that the commercial application is very closely associated to its application in nature. So that can give you a cue or an indication to what kind of commercial application a particular secondary metabolite may have. Now in conifers, monoterpene accumulate in resin ducts found in the needles and trunks. That is for what? Once it is in the bark region, then it can prevent either the pathogenic attack or even the herbivory. So many plants, they contain a mixture of volatile monoterpenes and sesquiterpenes, and these are called as essential oils, and they are highly expensive. So you will find that these essential oils are very expensive in market and they are not found in very large. I was looking for, we had gone to Coimbatore last month, and there in the shop I went to a tea plantation, where a factory where they were showing. So there was a shop in which they were selling these essential oils. So there was a tea tree oil which caught my attention. It was just 2 ml bottles which were being sold. So tiny one. And then I started asking what's the use of this. So they were saying it's a very expensive, very good oil for acne treatment, and then for, which means that antimicrobial activity and all. So by the time I was about to reach the counter to buy that tiny stuff, because that's what's very, I was asking why it is so expensive and why in such tiny. So they started telling me it's very difficult, the tree itself is not found, it's called tea tree oil. So that caught my attention because we work on plant biotechnology, so I thought it could be a good species to work with. So because the availability, the demand is high and the availability is less. So the minute by the time I could reach the counter to buy it, the stock got over. So they just had 2 bottles, and those 2 someone picked up before me. So that really has caught my attention, that essential oils are very expensive. They can have large commercial applications in cosmetic industries, in as your antimicrobial agents, so all related. So these are nothing, that was one example of essential oils. So they are frequently found in where? Glandular hairs, or so these are nothing but projections of the epidermis, epidermal hair, or even in trichomes. Trichomes are also found on the surface of the leaf, on the epidermis projections, where these secondary metabolites get accumulated in the form of bulbs. Now why do you think these essential oils are getting accumulated on the surface? That's the reason the plant will put it on the surface. How is insolubility related? The question was that why do you think these essential oils, generally you will find them on the surface of the leaf or on the surface, epidermis, dermal tissue near.
Protection from transpiration or something.
If it is volatile, it will help on the surface. Then if it is non-volatile and it has a role to play in attracting, so is only because of the, either the color should be responsible, that should also show up, or the odor should be responsible, that should also be on the surface. Then if it is acting as a defense mechanism, then should it be inside or should it be on the surface? First level of defense, so that, see, the plant is not moving, can move our hands and legs like us. So if at one point the attack is, it cannot move. It has to make sure that the rest of the parts of the plant are now geared to overcome the pathogen attack. So if the compound is on the surface, it will be first line of defense, it will be ruptured and then the secondary metabolite is released. So that is one of the reasons of having these in the form of trichomes or glandular hairs. Now triterpenes that defend plant against vertebrate herbivores include cardenolides and saponins. I don't expect you to remember, but at least I would like to tell you what are the different wide range of secondary metabolites, and they can have varied commercial applications. Now cardenolides are what? Glycosides. Now glycosylation, if you remember in the last classes, I told you addition of sugar moieties is sometimes to reduce the toxicity, one of the biotransformations. So glucosides. So even here cardenolides are glucosides. Now compounds containing an attached sugar or sugar alcohols that taste bitter and are extremely toxic to higher animals. So some of these glucosides, their taste is very bitter and which can act as a deterrent, feeding deterrent, or can be very toxic to the feeders, the herbivores. Now saponins are steroids and triterpene glycosides. So as I said earlier that there is wide range of secondary metabolism. So some of these intermediates can be directly getting produced to the final, used up in the final product in the straight biosynthetic pathway, but some may have one or more involvement of various more than one secondary metabolic pathways. Like for example saponins. Saponins are steroids and triterpene glycosides. So there is triterpene moiety and there is a steroidal moiety. So presence of both lipids, so which means what? It will be both water soluble, water insoluble. So it can form then micelles. So which can improve its diffusion through the membrane. Now I will talk about it later. Some of these secondary metabolites, how do you think the plant protect itself from the toxic action? It is said it is toxic. So one is they should be modified and stored, detoxified and stored, and when required, the required final step to again bring it back to the original, whether it is enzymatic step. So then the enzymes are stored in the vicinity, so that as soon as it is ruptured, and they are stored generally in suppose vacuoles. So tonoplast, for example, I said in this case, if it has to be stored in tonoplast, then in the form of micelles, it can easily pass through the phospholipid layer. But once it comes inside, then it is an acidic environment. So which will again transform and it will find it difficult to return back. So these are ways in which the plant would protect. But suppose this leaf where this was stored or stem part gets ruptured by the attack. So now this cell is ruptured, so everything will come out from the vacuole, although the cell has died, the one which is damaged now. But now whatever enzymes were there in the vicinity will also come, and the final step can take place with that intermediate and the enzyme, which can lead to the production of the final toxic metabolite. So it is not that it is very quick, and you will also find it is not for the very wound response by the plant, or the defense mechanism is not to have immediate sometimes defense, which means the cell which has died or the tissue which has been affected may not come back again, may not be revived like what we do as treatments, but the objective is to prevent its spread to the other parts of the plants or even to the neighboring plants. So that also forms a part of secondary metabolism. So the other class, as I said, was phenols. Now plants, they produce a large, so what do they have? They have, we know, a phenyl group attached with a functional group is what? OH. So that forms the phenolic group. Now these compounds, they are chemically heterogeneous, and almost 10,000 different phenolic compounds are produced as different secondary metabolites in plants. Now some are soluble in organic solvents, some are water soluble carbox in the form of, when they are in the form of carboxylic acids or glycosides, so they become water soluble, and some are insoluble polymers. So why do you think they are these different forms? And this is nothing but biotransformations, functional groups getting added, derivatives, saturation, unsaturation, which might change the chemical structure and its biological action in the plant. So phenolics play a variety of roles in the plant. What are the different roles? As defense against herbivores and pathogens. They can also act as mechanical support. Now I need to know, how do you think this phenolic compound can act as a mechanical support?
Probably since there are also polymers, large polymers which are present. So
Right. So they can also form a structural component of your polymers. Now these polymers in turn are utilized in your lignin, like for example lignin, it's a biopolymer. Similarly, other lines of defense where biopolymers are to be required. Now in attracting pollinators and fruit dispersers, now the colored pigments of plants usually provide visual cues that help to attract pollinators and seed dispersers. Now in absorbing harmful ultraviolet radiation, some of these compounds may also absorb the UV radiation. In reducing growth of the nearby competing plants. In vitro culture, sometimes we find that I referred to this, that some of the species, because of large exudation of phenolic compounds, it becomes very difficult to grow them in vitro. That is because of the toxic nature of the phenolic compounds which get released in the medium. So phenolic pigments are of two principal types. One is carotenoids and flavonoids. Now carotenoids, these are isoprenoid alcohols. For example, there are different colors, yellow, orange, red terpenoid compounds that serve as accessory pigments in photosynthesis. So it is not only green color or your chloroplast. Sometimes you will find that there is a transformation from chloroplast to chromoplast and lycopene. So sometimes in vitro cultures, in order to enhance the production of, there is an example of lycopene, in some elicitors were added, but it was also observed that once the lycopene, so it is works the other way around, backward direction. Once the lycopene synthesis started happening, the chloroplast started getting transformed, reduced, and the proplastid, which is a previous stage of forming different types of plastid, they started getting converted to chromoplast. So that also is nothing but, now these sometimes far UV range, which is a lower wavelength, which are not visible to the eye, but some of these secondary metabolites can capture light energy which is in this shorter wavelength area. So the flavonoids also include a wide range of colored substances. The most widespread group of pigmented flavonoids is anthocyanins. These are commercially very important molecules, secondary metabolites. Now which are responsible for most of the red, pink, your purple colors of the fruits and flowers. Now the two other groups of flavonoids that found in flowers are flavones and flavonols. Now these are those secondary metabolites which are also used not only for the helping in pollination as or they have other defense related activities, but they can also help in light absorption because they can absorb light energy in the shorter wavelength. So isoflavonoids, which are found mostly in legumes and have several different biological activities. Rotenone can be used effectively as insecticide or for pesticide, which is for killing the fishes. So isoflavones may also be responsible for the anti-cancer benefit of food prepared from soybeans. Now phenolic polymers, now these phenolic polymers, they have defensive properties besides lignin and are called as tannins. So that is what we may say that they are utilized in the formation of defense. Now they are general toxins that can reduce the growth and survival of many herbivores. Now tannins, they act as feeding repellents to great variety of animals. Unripe fruits, for example, now the plant would not like anybody to eat the unripe fruit until unless the seed is ready for the next stage. So it then produces more amounts. Where will the plant produce more amount of such compounds? It will produce inside the fruit so that it can deter the herbivores. You will find that I also spoke about that azadirachtin in the plant is found maximally where? In the seeds. What can be the reason? So as to prevent, it will act as a feeding deterrent. It is bitter in taste. You will find neem leaves are very bitter and the seed is also very bitter, but the ripe seeds, they are full of sugar. So the release of secondary compounds by one plant that have an effect on neighboring plants is called allelopathy. If a plant can reduce the growth of nearby plants by releasing chemicals into the soil, it may increase its access to its competition. So it would like to reduce the growth of the nearby plants. So secondary metabolism also plays a role there. Now, how do you think that in all this the plant will protect itself? One is the storage. What else? All the compounds will be stored. There are so many compounds which the plant is preparing. Vacuole, so how much can vacuole hold? Thousands are getting produced. So everything is not getting stored in just vacuoles. Some are in the open spaces also in the plant, some particular tissue, or you will find that it is in cortex cells directly accumulating there. You can visualize them in a tissue section. But then how do you think the plant protect itself? If suppose it is acting, let us take camptothecin, because we are studying camptothecin, so we know about it. So in that I will take that as an example. So camptothecin, it is a topoisomerase inhibitor, that is why it is a very good candidate for anti-cancer activity. So it is cytotoxic in nature. Now it is also present in plants, so then how do the plants protect itself? The topoisomerase of the plant gets mutated such that the mode of action, that is why I said, see, the mode of action in cells, in mammalian cells, else is the topoisomerase is different than in the plants. So that is how plant protect itself from the cytotoxic effect of camptothecin. So alkaloids, so a large variety of the plant secondary metabolites, they have nitrogen as a part of their structure. So nitrogen containing organic compounds, they form alkaloids. Now the well known anti-herbivore defense, hence these are called as alkaloids. Now most nitrogenous secondary metabolites are synthesized from common amino acids. So most of these alkaloids, they are formed from amino acids. So alkaloids are usually synthesized from one of the few common amino acids. What are these? Lysine, tyrosine or tryptophan. The alkaloids are a large family of more than 15,000 nitrogen containing secondary metabolites. So now can you see 10,000 phenols, 15,000 alkaloids, and the largest group was triterpenes, much more than that. So imagine the wide array of secondary metabolites the plants are producing. So how much carbon and energy is getting utilized apart from growth into secondary metabolites. So no wonder the plant has to judiciously prioritize the carbon flux into these wide array of secondary metabolites. What else do you think? How does the plant prioritize then? The enzymes which are involved in the biosynthesis of these, they in turn get impacted by these external factors, maybe temperature, light or elicitors. So that is how the plant is all the time managing its resource. Maybe the enzyme is there, maybe enzyme is not at all there. We will see it later as I go on, that there are a class of secondary metabolites which are called as phytoalexins. Phytoalexins are not present, they are synthesized de novo when needed, but there the trick is the synthesis is very fast, and there are constitutive secondary metabolites which are present all the time. So these are different ways in which the plant is prioritizing.